है मेंडेलियन डिसऑर्डर। इससे बहुत सारे क्वेश्चंस पूछते हैं, 1 वर्ड क्वेश्चंस, 2 मार्क्स क्वेश्चंस, या इसका कहते हैं कि सिम्टम्स एटसेट्रा, कॉज। इसलिए इसको एक नज़र में 5 मिनट के अंदर आराम से इसकी तैयारी हो सकती है। तो मेंडेलियन डिसऑर्डर, दिस टाइप ऑफ डिसऑर्डर इज ओनली ड्यू टू mutation or alteration in a single gene, It is also called point mutation. Means any kind of change or either deletion, addition, subtractions, etc. in a single gene, this cause such type of disorders. This is called Mendelian disorders. And one of the important disorders, that is Haemophilia. It is also called bleeder's disease. It is sex-linked recessive disease. Means the gene of this disease is located on a sex chromosome, that is X-linked disease. It is all not sex-linked, you may say. This is X-linked disease. From this disease can be transmitted from carrier female to normal son. And a single protein essential for blood clotting, protein cascade, is affected. Means the proteins involved in blood clotting, which forms a cascade mechanism in which one single protein is affected, and due to lack of this protein, this bleeder's disease occurs. By the following cross, we can explain it. Suppose a normal father is crossed with carrier mother. The normal father, genotype is XY, genotype is XX. This forms XY gamete, this forms XX gamete. When this sperm fuses with this ovum, it gives XX, this normal daughter. When this sperm fuses with this ovum, then produces carrier daughter. When this sperm fuses with this ovum, this normal son, and this produces with XY affected son. As we see in this cross, father is normal, mother is carrier. So all the 50% of daughter is normal, 50% is carrier, and 50% son is normal and 50% is affected. As I have written here, from carrier female to normal son. Normal son is affected. In this way, if father is, you can complete this cross. Father is affected, mother is normal, one cross, another cross. Father is normal, mother is affected. If mother is affected, then the gene of hemophilia will locate on both the chromosomes. And the third category, as I have explained in the above, crisscross mechanism, that is, father is normal, mother is carrier. In this way, hemophilia can be explained. Now the next Mendelian disease is Sickle-Cell Anemia. It is autosomal linked recessive disease. As we know, hemoglobin has two types of chain, alpha chain, beta chain. And the normal hemoglobin, this is the symbol of hemoglobin, has glutamic acid on 6th position of beta globin chain. This is the characteristic picture. This is very important point. 6th position. This is very important. Which amino acids? Glutamic acid. This is normal. But in sickle cell anemia, what happens in hemoglobin, sickle cell hemoglobin? This is the symbol, and this is the normal hemoglobin. This is the sickle cell hemoglobin. This glutamic acid is replaced by valine. Due to this reason, RBC gets low O2 tension and gets changed into sickle shaped from biconvex, from biconcave shape. Means normal has either biconcave shape, तो इसकी वजह से ये सिकल शेप में चेंज हो जाता है. इसका वन ऑफ द फैक्टर है लो ऑक्सीजन टेंशन. आरबीसी चेंज हो जाता है जिससे ऑक्सीजन की सप्लाई कॉजिंग लो सप्लाई ऑफ oxygen and bleeding, suffocation. So these underlined words are very important, and you must underline when you write the answer. This is the key point. Now we can explain with this help of this HbA. This is normal hemoglobin genotype. HbA, HbS carrier, HbS sufferer. And suppose a normal man is crossed with carrier woman, then HbA, this is the gamete, and HbA, HbS. When this fuses with this, HbA, 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 HbS, HbA, HbS. This is the carrier, normal, means 50% is carrier and 50% is normal. Now the next disease is thalassemia, PKU. The next disease, this hemoglobin or sickle-cell anemia can also be explained by this graph, and it is generally it is asked. Normal HB, this is CTC, C combines with G, T combines with A and G. This is gene. A gene. mRNA, synthesis of mRNA. It gives GAG. It gives valine 1 linked with histidine 2 linked with leucine 3 linked with threonine 4 linked with proline 5 linked with glutamic acid 6 and then glutamic acid. So this is the normal gene, normal. So this sixth position, in place of this, here instead of glutamic acid, here is valine. Here is valine in HbS. Instead of, and here, instead of, see, this is the HbS gene, CACGTG gene, and then here is mRNA, the GTG, GTG, here is GUG, here is, and then here is at this position the same chain is here, only in place of glutamic acid there is a valine. Now the next disease is PKU, phenyl ketonuria, और phenyl ketonuria. It is inborn error. Inborn error means पैदाइशी of metabolism. Inborn error of metabolism means there is some problem or error in the converting problems. What is problem? It is autosomal recessive disease. Normally, phenylalanine, an amino acid, is converted into tyrosine. With the help of enzyme phenylalanine hydroxyxylase. In this disease, this enzyme is lacking, causing conversion of phenylalanine into phenylpyruvic acid, which accumulates in brain, cause mental retardation. This is the key point. Phenylpyruvic acid is the key point. Phenylalanine is the key point. This phenylalanine, tyrosine, normally हमारे बॉडी में क्या होता है कि फिनाइलएलेनिन जो है टायरोसिन में कन्वर्ट हो जाता है. अगर ये एंजाइम मौजूद रहता है तो इस बीमारी में क्या होता है कि ये एंजाइम लैक हो जाता है, कम हो जाता है या खत्म हो जाता है, जिसकी वजह से फिनाइलएलेनिन टायरोसिन में कन्वर्ट नहीं हो पाता है और ये फिनाइलएलेनिन पायरुविक एसिड में कन्वर्ट हो जाता है जो कि ब्रेन में जाके जमा हो जाता है, जिसकी वजह से मेंटल रिटार्डेशन होता है, यानी दिमाग कम ग्रो करता है. द नेक्स्ट वन इज नेक्स्ट डिजीज इज कलर ब्लाइंडनेस. It is CBS, simply called CBS, color blindness. As the name suggests, color blindness means people are not able to distinguish between colors. Means लोग जो है इस बीमारी से जो ग्रसित होते हैं, जो सफर्ड होते हैं, वो रेड और ग्रीन कलर में डिफरेंस नहीं बता पाते हैं. सेक्स लिंक्ड डिजीज, रेसेसिव डिजीज. Important point. Person fails to distinguish between red and green color. In this disease, gene for normal vision is dominant. If gene is located only one X chromosome, then it is called carrier. Gene is located both the X chromosome, then affected. And in case of male, only X-linked, where it is better to write X-linked sex-linked recessive disease, X chromosome, that is affected. Now, suppose affected father is crossed with carrier mother. Mother is carrier. The genotype of affected father is XY, having gene, carrier, X gene. Now the gamete is XC, Y, here in the says X and X. If this sperm fuses with this X chromosome, then the is like this. And if this sperm fuses with this, then like this. This sperm fuses with this XY, and then this XC. So here there are 4 progeny. Out of 4 progeny, by including male and female, 50% are normal, 50% are affected, 25% are normal, and 25% are carrier. So 25% carrier, 25% normal, 50% affected among all 4 children, 4 progeny. By another cross, you can explain it. Now the next disease is thalassemia. Next disease is thalassemia. It is also autosomal linked recessive disease. It is caused due to defect in synthesis of globin chain. It is 3 types. Number 1, alpha thalassemia. Two gene HB1A1, HBA2. These 2 genes are located on 16th chromosome and of alpha chain. 16th chromosome is affected. In this case, alpha chain is affected. Now the next one is beta thalassemia. Beta thalassemia, as the name suggests, beta chain is affected. HBB, this is the gene causing beta thalassemia, located on 11th chromosome. Now the next one is delta thalassemia. It is rare and it is due to delta chain is affected and gene is HBD located on 11th chromosome. Now, difference between sickle cell anemia and thalassemia. This is important. Sickle cell anemia and thalassemia. No inequality in the synthesis of alpha and beta chains. One chain in a small quantity and another synthesized in large quantity. RBC becomes sickle cell, sickle type shaped. Globin attaches to RBC membrane and damage it, and damage the same. Defected chain forms helical hemoglobin strand of excess chain forms PPT. Sickle cell anemia it is qualitative and it is quantitative. I hope that it will help you in the forthcoming board exam. Thank you.